So it's my pleasure to introduce Doctors Kollman and Amenyogbe from the Telethon Kids Institute in Australia. And to let you know a little bit about them, they both moved from Canada, actually, their last place to out in UBC in Western Canada, to the Perth Telethon Kids Institute. Dr. Kollman has had a long-standing interest in childhood diseases and early childhood sepsis and so on. He has got his MD and his PhD from the Albert Einstein College of Medicine in New York, and subsequently did a fellowship in infectious diseases at the University of Washington in Seattle. He was division head at pediatric infectious diseases, as I said, at UBC, University of British Columbia in Canada, and currently is professor of systems vaccinology at the Telethon Kids Institute in Perth. He's also a director of systems immunology with the Human Vaccine Project, which is a global not-for-profit network which aims to decode the human immune system. He's very interested in newborn infectious diseases, immune ontogeny, and early life vaccine responses. And he's going to talk a little bit about that today. And Nellie Amenyogbe is a PhD research fellow, also at the Telethon Kids Institute, whose expertise is also related to understanding the host's immune fitness, with a special interest on global infectious diseases, and focusing again on system immunity, the gut microbiome, and an interest in children around the globe who are vulnerable to newborn sepsis, and examining how she and her team can address these issues. So rather than cut in any more into their time, I'm going to welcome them both, friends and colleagues, and invite them to give their presentation to us today on why do newborns die? A paradigm shift from pathogen to the host. Over to you.
All right, thank you so much, Eleanor. Let's just get our handy screen share. Fingers crossed this time around there's going to be no glitches, and we've had our fair share of experiences. What a pleasure to be at Beyond Sciences. I've attended this conference a few times. I've won a prize for presenting at this conference, and one of my students from Guinea-Bissau has also won a prize from presenting at this conference, and she was very proud of that. There you go, now we're good. So it's always a pleasure to join everyone here. And it's wonderful how international this all is. Toby's going to start the presentation, and then I will carry on about halfway through. So I'll hand it over for now, and I will see everyone in about 2 minutes.
All right. Well, thank you, Eleanor, for the introduction, and thank you all for attending this great conference. We're going to indeed talk about why do newborns die, and introduce a paradigm shift away from a focus on the pathogen, not to ignore the pathogen, but towards the host, to emphasize host resilience. What we will show today is 4 parts, namely part 1, the problem of newborn infection, then part 2, part of the solution from an old vaccine, part 3, looking ahead into the future, reframing newborn host defense, and part 4, really looking far ahead and introducing an approach where we actually ask the newborns to tell us where to go with effective interventions. And at the end, we're also going to introduce some exciting opportunities for anyone interested for either a PhD studentship or a postdoctoral fellowship. Part 1, the problem of newborn deaths. This is the world represented graphically with newborn deaths per 1,000 live births. And as you can see, the vast majority of newborn deaths affect lower and middle income countries. For many of you, that's not a surprise. Many of you live in these areas, but it is a daily burden that hits low resource settings the hardest. Newborn mortality makes up over 50% of all deaths under 5 years of age, meaning more than half of all childhood deaths happen in the first month of life. And approximately half of those are actually due to infection. There's multiple pie charts that I'm sure some of you have seen in the past. This is one of the most recent and also one of the most comprehensive ones given to us thanks to Shabir Mahdi, who's part of the CHAMPS team that uses post-mortem biopsies, minimally invasive biopsies, to identify using molecular tools what the cause of death was. And they confirmed over 50% or close to 50% of all deaths in the newborn period are actually due to infections. More specifically, the microbes identified using these molecular technologies, not just blood cultures, range far and wide. Viruses such as RSV in this graph, and the bar graph represents the relative proportion of pathogens identified in this complex workup that I just mentioned. But what really shocked most of us was that the number one pathogen identified was urea plasma, something that most physicians don't even consider pathogenic, but there it is. So we're missing something. But the most important finding from all of these studies was that over 70% had no pathogen identified. Sure, many of these have been missed despite this elaborate setup. But it could also argue that what really kills newborns and appears to be an infection clinically wasn't really an infection. There's other mechanisms. This has been already proposed decades ago, but now we're starting closer to get to the truth. Can vaccines help? This is a graph of studies conducted in Southeast Asia on the right, South Africa and Southern Africa and Sub-Saharan Africa, with a large number of participants from 6,000, 1,000 to, in Sub-Saharan Africa, over 84,000. Identified here as a representative bar graph in this spiral plot is the relative contribution of identified pathogens, and labeled in orange are the pathogens for which we could potentially have a vaccine available, although they're currently not used in the newborn age group. And you very rapidly see that the vast majority of pathogens identified as killers in early life do not have a vaccine even available, even if we wanted to. And what is even more important is when you consider when newborns die. This is a graphical representation put together by UNICEF and the WHO of all the causes of death that have been identified in the newborn age group. By far, the biggest cause of death is related to preterm birth, and I'll get back to this, perinatal or intrapartum related complications, and then sepsis, so a very severe form of infection. If you just focus on sepsis alone, and you look at when do these newborns die from a severe form of infection, the vast majority dies in the first few days of life. Not so much, half of them then in week 2, and then even less than that over week 3 and 4. So the vast majority of deaths due to severe infection in the newborn age group happen in the first few days of life. The newborn period is typically captured as the first month or up to 28 days of life. But within that period, the newborn deaths concentrate in the first few days of life. If you were to have a vaccine, you'd give it at birth. It takes on average about 2 weeks to even induce an immune response. So even if you had vaccines for all these different pathogens that I just showed you before that have been shown to be associated with newborn deaths due to infection, even if you had those vaccines and even if you would give them on day 1 of birth, we would fail to stop this onslaught of killers. So what this really means is the way we currently approach to protect more than half or stop more than half of all newborn deaths is simply not working. And it will not work. It misses the actual cause of death because the pathogens that kill are not covered by vaccines as we currently approach them, and it misses the timing. So really, the current approach to protecting newborns from infectious death is not going to work. Vaccines alone, they help, especially maternal vaccines help, but they will not suffice. Pathogen-specific approaches, one vaccine targeting one pathogen is not going to do the trick. We reviewed this about 2 years ago for Science in a special edition that's cited here. What also is important to point out here, as I mentioned this already, that preterm birth is the single largest cause of neonatal deaths and also the major cause, major risk factor for newborn infections. Preterm birth most hardly hits lower resource settings, but it also is one of the leading causes of child death in high-income countries, whether it's Australia, Canada, or the US, or anywhere in the European countries. Preterm birth adds up to about 15 million per year. And it's increasing, not decreasing. Currently, anywhere from 5 to 20% on average, about 10% of all pregnancies end in preterm birth. And this is on top of stillbirth, which is currently not well captured, but estimated to happen at around 2.5 million deaths per year. And to put this into perspective, as Joylan has done recently during our Keystone Conference on this topic, together, stillbirth, preterm birth, and the consequences, most particularly neonatal infections, together represent the longest standing, deadliest pandemic of human history. And we all have seemingly grown accustomed to this, accepting that this is the status quo. It is unacceptable. It is unethical. It is immoral not to make this the priority of our undertakings. To this end, we've started the Born Strong initiative. It will be fully rolled out sometime later this year, but in the Born Strong initiative, we're targeting interventions that have been shown, have been proven with promise to interrupt this spiral of preterm birth and with that newborn infections. And we use existing interventions such that they don't have to be licensed again. We're not talking about something that's going to have impact 20 years from now, but 2 years from now. So stay tuned to what's going to come with the Born Strong initiative. Already proven as a solution to intervene in this is something that Nelly will introduce to you now as part of the solution. And with this, approaching part 2 of this presentation.
Oh, hello, everybody. Oh, this is one of my favorite photos, and I love to use it for a few lectures. It's beautiful and serene. Green has a natural calming influence. But more importantly, what we're looking at is a picture of a vaccine clinic. This photo is taken in Guinea-Bissau. And if you looked at the side of this white truck, you would see a little sign for a group called the Bandom Health Project. And the sign here would actually be in Portuguese, which is the local language. But this is really the foundation of where a lot of our research has actually been focused over the last 10 years, slightly over 10 years. But a story that's not nearly as known as it should be, which is why I like to start most of my talks around the BCG vaccine and how we came here from Guinea-Bissau. So I like to kind of scatter my acknowledgements throughout my slides instead of putting them at the very end. Lets people know where the contributions came from. And here, a lot of the credit would go to both to Peter Abbey and Christine Stabell-Ben. A few of you might have heard of these people before. And also here's pictured Byron Brooke, our former PhD student, who is one of the hardest working people that I've ever met, and was responsible for a lot of the most work that I will share with you later today. But Peter Abbey has been working in Guinea-Bissau since the late 1970s, and he's still there. He's been joined by Christine Stabell-Ben for many of those years. And together, a lot of them, they have uncovered some very important epidemiological phenomena that if only us immunologists were to listen to anthropologists more often, we might get further in a science a lot faster than if we just stayed in our labs and looked at our immune cells only. So this is Guinea-Bissau. This photo was taken not so long ago, I think around 2016. It's a former Portuguese colony. It gained independence in 1974. The path to independence was a struggle for Guinea-Bissau, as it was for many African nations. But what happened here is it led the country in a lot of turmoil, to the point that in the late 1970s, the infant mortality rate that was 500 per 1,000 person years. So about half of children that died didn't survive their childhoods. And obviously something had to be done about it. And that something was to dispatch a team of Europeans, including Peter Abbey, to Guinea-Bissau to start the first vaccine rollouts in the country. Now, they didn't travel there to find out what they did find out, but as good anthropologists do, they collect data and they look at it in unbiased ways. And this is really how the BCG story came about. So when they started to roll out these vaccines in Guinea-Bissau, first the measles vaccine and later on BCG. The BCG is the vaccine that we give against tuberculosis, and everyone that's got a little scar on the side of their arm, likely most of you are probably too young to have been vaccinated with smallpox. Probably it was the BCG vaccine that you received early in your infancy. And what they noticed was that the newborns that received BCG were less likely to die than the ones that hadn't received BCG, even though tuberculosis A is not a major cause of death amongst newborns, but B, it wasn't really circulating amongst the population at all. And so instead of brushing this finding away, they wanted to lean in and look at it further. And they were able to explore that in a randomized control trial, because normally low birth weight newborns, so mostly preterm. Of course, here term low birth weight because gestational age is not often assigned. Get vaccinated with BCG when they're about a month of age. But of course, as you heard from Toby before, most of the mortality that happened would have happened earlier and thus was completely missed at that time. So they performed this randomized trial to see if babies that were vaccinated at birth would benefit compared to whether they would be vaccinated a month later. And sure enough, they what they found was promising in that there was a 50% reduction in all-cause mortality amongst newborns that were vaccinated immediately compared to ones that were vaccinated a lot later. And that's massive. That's all-cause mortality. That's not one pathogen, and each one of those pathogens accounts for a tiny fraction of total cause of death. So you would think that the world would go crazy about this and actually start to capitalize on this in order to do something about newborn sepsis, but it was quite the opposite. It was met with a lot of skepticism, and here is the response to their publication saying that because this tendency appeared only 3 days after BCG vaccination, it seems unlikely that an immunological mechanism could explain such rapid impact on mortality. So of course they pressed on, and since then other confirmatory trials have been done, and a review has been commissioned by the World Health Organization. And all in all, it was deemed plausible that this was happening, but because there was no known mechanism, there was still some skepticism around that. And because of that, we haven't really done much about this in terms of policy or implementation. We still think that BCG is only a vaccine against tuberculosis. We don't use it in ways which recognize the other effects that it has. So as immunologists around 2012, we formed our own hypothesis around this, that this is a very interesting finding. And we thought, could we actually find out more about this by researching it into a laboratory? So that's where our hypothesis came from, that BCG's non-specific protection must be at least in part accounted for by a very rapid change in newborn immunity that affords protection from overwhelming infection. And so we designed an animal investigation to do this. We chose a polymicrobial sepsis model that was developed by one of our collaborators, a neonatologist working at the University of Florida. We vaccinated our mice with BCG, the same BCG that was given in Guinea-Bissau, and they were challenged only 3 days later, mostly to mirror the kinetics that were noted in the original publication. This is important because timing is very important, and this is why we looked closely at what the kinetics were in the original observations before we designed our mouse studies. And of course, we saw a beneficial effect. So BCG vaccinated mice did better. Their risk for survival was, their chances of survival were improved by about 40%. They had a lower inflammatory tone. So here, for example, we're showing TNF alpha, one of the prototypical cytokines that we look at when we look at the inflammatory response. And when we looked at all of their organs, we found that they had also cleared their bacteria faster compared to the control mice. When we looked under the hood, the cell type that we saw that was most profoundly impacted by BCG prior to challenge was the neutrophil compartment. So what we saw was the total neutrophil pools in both the bone marrows and the spleens of these mice expanded about 3 days after BCG vaccination. This was true for both immature and for mature neutrophils. And then after the mice were challenged, these neutrophil pools were quickly depleted and were essentially recruited from the sites where they were sitting, so the spleen and the bone marrow. And we hypothesized contributed to this enhanced clearance. Now, expanding your neutrophils doesn't seem like anything fancy to do, but for a newborn, and I will touch on energetic constraints for the newborn period later on, it is a big deal. It takes a lot of resources to expand cells, and this process for our bodies is called emergency granulopoiesis. It's driven by a cytokine called granulocyte colony stimulating factor, GCSF. It was induced within hours following BCG, and then 3 days later is when the neutrophils showed up. So when we blocked GCSF, we saw that we also blocked the expansion of neutrophils as well. The other data piece that I'm not showing you is if we adoptively transfer those neutrophils in the equivalent numbers from BCG vaccinated mice compared to unvaccinated mice, we saw that the doubling of neutrophils also accounted for the rate of bacterial clearance that we observed. In our earlier work, we also established that bacterial load was heavily linked to risk for survival in our mouse model as well. There's many situations in which bacterial burden or viral burdens might not necessarily link to host welfare. So we did have to do our homework and check that that was the case. But we also looked back to human cohorts. Now we'll touch on this cohort a little bit later in today's talk as well. But through the expanded program of immunization consortium, or EPIC, we were able to quantify the number of neutrophils in BCG vaccinated and unvaccinated newborns at 1, 3, or 7 days post-vaccination, found that at 3 days specifically, the vaccinated newborns had an increased number of circulating neutrophils. So to put the picture together, we found that in normal conditions, your body runs a process called steady state granulopoiesis. You don't have a need to ramp up your neutrophils all the time. And so you don't. You use your energy for other things. However, for newborns that are at higher risk for sepsis, that need to clear pathogens very quickly, and that are often neutropenic to begin with, especially low birth weight newborns, having more neutrophils on board is a great advantage, as is what happens after BCG vaccination, where in the spleen, you have an upregulation of a transcription factor called CBP beta. It drives up emergency granulopoiesis. These neutrophils are not doing anything harmful in the body. They're just sitting and waiting. But when we challenge the mice with bacteria, they're very quickly deployed and are able to increase their risk for survival in our mouse model. And after 8 years of work, we were finally able to publish our findings in 2020, so 2 years ago. And what grew from a project that was really involving maybe 1 or 2 people ended up being a massive team effort involving people from all over the world. So it goes to show you how the world can come together to actually make a difference and deliver some important findings. And all of this was really because some epidemiologists in Guinea-Bissau made an observation and hung on to it. They just didn't let it go. So mystery solved. Well, the non-specific effects of vaccines are being increasingly recognized. This isn't only BCG, but for pretty much all vaccines that we give have some non-specific effects. Some of those are very powerful and really need to be looked at more closely. Many questions do remain around BCG vaccine outside of the newborn period. Of course, BCG has had a lot of focus, even in COVID, and people have been exploring if BCG could be used to reduce lower respiratory tract infections later in the life course. But for us, because we're still focused in the newborn space, what's important is BCG doesn't always work. And to understand why, we need to understand immuno-ontogeny as well. This is where we go into part 3, reframing host defense. So there's 2 main things that we're now researching in a lab. One is, why doesn't this work for all newborns, including in our mouse model, by the way? It's not effective 100% of the time, and we don't have a good understanding of why. And we also actually don't know exactly how BCG induces emergency granulopoiesis. In short, we've tried mouse knockouts for every receptor that should have a role in recognizing mycobacteria. And none of that is enough to knock off this process. So that's a big question mark for us as well. But going back to the first one, why doesn't it work all the time? Well, once we started looking at BCG, we started to look at sepsis, which is not something that a lab was researching at the time. But when we brought in this model, we realized that we actually had a very poor understanding of why newborns died of sepsis to begin with, especially when we couldn't find pathogens most of the time. And when you started thinking about this more, we very quickly realized that as immunologists, we were really going astray by not making the host metabolic state a central focus of our research. This was around the time when we really stopped calling the newborns immature and realized that their immune system was not immature. It was custom tailored to what the newborn needed to do. And the newborn is in a state of a very high energetic demand with few choices of what kind of immune response to deploy. So there has been a great literature on the cost of infectious diseases, and most of that has been done for adults, where we know if you have a given amount of energy, normally you're responsible for your homeostatic processes, unless you're in growth and reproductive phases. But if you have an infectious event, you've got a lot of energy that you can dictate to the host response. So you might choose a strategy of disease resistance, for example, but not if you're a newborn. You might not be able to afford that. So you constrain the types of things you're able to do towards disease tolerance instead. And so the things that work for newborns have to work in that context as well. This concept was beautifully explored quite recently in a paper here on energetic trade-offs, where in response to bacterial or lipopolysaccharide challenges, mice orchestrate and they go into a hypometabolic state, where they drop their body temperature and drop their drop their metabolic rate and reshuffle those resources to the host response. So we started to look at this in our newborn mouse models as well. Here I have pictured my team here in Perth. So Adrian Eno, our former master student, Joan Solomon, and one of our research fellows, Elizabeth Schumann, who do what needs to be done in order to run a time course that hits all of these time points. So of course, we've got people in the lab sometimes at 2 o'clock in the morning to make measurements and check on our mice. And what we see in the early times after challenge is mice that survive ramp up their metabolism very slightly, something that's indicative of switching over to burning ketones instead of fats is what they were doing before. But what we also see is that mice that are vaccinated with BCG, and this is pilot data only, but they tend to exit from the state a little bit earlier. It doesn't seem like this happens for all mice, however, and that goes into our lack of understanding of why that happens. We don't know why some mice don't exit out of this hypometabolic state. Likely has to do with their fueling. Which brings us to the next part, which is where we now have to go back and ask newborns to tell us where it is that we need to go with our research instead of thinking that we know the way forward. And this is where we got into systems biology. So we did have a chance to actually start collaborating with Christine and Peter in the Bantam Health Project. And to do that, we had to master getting massive amounts of data out of very tiny samples. So this initiated, we initiated a study in 2015 where we sampled newborns that either received BCG or didn't, and we looked at their in-hospital outcomes. Out of only 400 microliters of blood, we were able to capture their cellular composition, their plasma proteomes, metabolomes, and their whole blood transcriptome. So really a massive amount of data. And while that study was going on, we actually had the opportunity to now take this protocol and adapt it to the expanded program of immunization consortium, also focused on newborn responses, newborn immune ontogeny, focused around vaccine responses. So I will show you a little bit of data from that a few insights that we've gotten from this cohort, but really a massive team effort. I played a small role in this as well, but just goes to show that when any of these endeavors come together, it takes a lot of really talented folks and a lot of knowledge in order for this to work well. And so this is where our segue into systems biology gets. Even from the years that I've been in immunology, so I started my graduate training in 2012, I've seen the field change massively from looking at little things at a time, picking a few cytokines and tracking them, to really taking advantage of systems biology to point the lens at very different systems in order to get a more complete answer. And this is what EPIC decided to do as well. We use data integration to find signals of immune ontogeny, and in short, we were really able to take a deep dive into the first week of life, and we could characterize with great accuracy the day of life of which the number of days that the newborn was alive based on what was happening in their bloodstream. Really amazing, considering that if I was to sample an infant for a week or an adult for a week, you would never see such a rapid change as you do in this early period, yet we understand very little about this. And with that, we can't really use that knowledge to our advantage. So this was also published, and again, it kind of gives a sense of how many people are involved in these large endeavors. This came out in 2019. And of course, now we're carrying this platform forward in order to now start to understand what differs in disease states as well, including in the BCG immediate study. Before we end our talk, we wanted to plug one small initiative that we hope will be a big initiative in some time to come, and that is access to knowledge that allows teams to access systems biology pipelines, because data science is for everyone. And one of the biggest things that I learned in my graduate training is fluency in data analytics makes me a much better scientist and gives me the power to really understand my data a lot more. So data literacy is becoming an essential skill for scientists across all disciplines. But it's tough. It's tough to get started, and you don't know where to start or how to start. We don't know how to coach our students, and we don't know how to make this accessible. So this is why we are, along with our colleagues here, Rim Ben Othman and Rad Aniba. Rim is also on one of our former slides, heavily involved in EPIC. We are putting together something called the Virtual Academy. It's going to be a global online platform where you can access knowledge on how to work with data from basics in our programming all the way through to systems biology and data governance. I will put a link in the chat right after our talk is done for a short survey that we're deploying. If you're interested in this, put your email down. So when we're ready to go in later in this year, we can get in touch with you. So in summary, efforts to tackle newborn infection are not sufficient. We need to start thinking outside our boxes and start to think, start to reframe how newborns develop and start to stop calling them immature as well, and start to admit that we really just don't know as much as we think we do, which is why we need to rigorously apply systems biology. Our team in Perth is growing. In the near future, we're always looking for people that are already passionate in this field and want to contribute to work with our global team. And with that, we are happy to take any questions. I can just stop my screen share.
Thank you very much, Nellie and Toby. That was a really exciting talk, and I think it captured what the issue is and opens the door to how we tackle this. So there have been a few questions, but I'm going to take the liberty of asking the first one. So RNA vaccines, nobody's talking about, well, very few people are talking, but nobody's considering the off-target effects of RNA vaccines. Have you considered that in the context of the kind of work that you've been doing, and what might be the impact, not even considering adults, but perhaps we know that pregnant women are receiving these vaccines, and we know that children as young as the age of 5 are. Any thoughts on what would be the off-target effects?
Yes, so the short answer is we don't know. So we only have thoughts at this point. What we do know is that vaccines, the COVID vaccines that are currently given to women during pregnancy, overall appear safe. There's no obvious signal at this point right now that indicate that there's any increase in adverse pregnancy outcome. The immune response is also overall equivalent. So it doesn't seem that administering RNA vaccines for COVID to women during pregnancy has any measurable downside. So far as we can tell, and that's obviously not very much in terms of length and duration of observation, that the newborns that are the product of these pregnancies where women have been immunized with the RNA vaccines also appear to be overall not significantly different in terms of risk factors for infection or other outcomes. Again, this is not any long-term follow-up, so we don't really know enough about this. Having said that, we do know from other studies that vaccinating the mother during pregnancy can have a substantial impact on newborns. Particularly, for example, BCG, as the vaccine that Nelly was just talking about, in observational studies, or not in mechanistic studies, has been shown to reduce the risk for adverse pregnancy outcome, preterm birth, or more specifically low birth weight or very low birth weight births on a population level. Similarly, at least in Victoria and in the eastern states of Australia, immunization of the mother during pregnancy with pertussis or RNA or tetanus vaccines also decreases adverse pregnancy outcomes. So immunizing the mother may have impact on the newborn more than just targeting the pathogen. Getting back to the RNA vaccine specifically, if you administer the RNA vaccines to newborns, and I trust that that will happen not so long from now, that the vaccine's eligibility is going to be broadened. The likelihood of there being potential risks isn't small. One of the major risks that drives newborns to be more symptomatic from infection actually relates to type 1 interferon production. If they are induced to produce type 1 interferon, and Eleanor is one of the world's experts in this field, if newborns are induced to produce type 1 interferon in response to infection, they often cause a massive amount of harm. So the risk of hyperinterferon induction in newborns, especially via the STING pathway, is substantial. With that, I would anticipate that the progress towards immunizing newborns with RNA-based vaccines has to be done very cautiously and very carefully and very slow, which would be the ideal approach. Whether that's what's going to happen or not remains to be determined, because this pandemic has thrown many a curveball at all of us, and sometimes making the things move faster than would have been preferred. So we don't know enough. I think this will be an issue that needs to be addressed sooner rather than later. I anticipate that immunization during pregnancy will show itself to be reasonably safe. I anticipate that immunizing newborns with RNA-based vaccines may have problems.
Okay, one of the questions from the Q&A is, have you looked at the durability of antibody and T-cell responses with BCG in newborns?
So, if I may?
Yeah, go ahead.
So this has been done, obviously BCG has been around for 100+ years. And there's been study after study that has looked at this, and it is very interesting that if you immunize newborns with BCG, if you immunize with BCG around birth, the immune response that you induce is as durable, as broad, and as potent, if that's the right word, in terms of quantity of cytokines and the likes that are being induced, as when you immunize older children or adults. So there is no clear downside to immunizing with respect to protection from TB, which is what BCG originally was designed for, immunizing at birth versus later on. The interesting part is that immunizing at birth seems to in fact be a better way to protect from tuberculosis than immunizing later in childhood or in adolescence. So there is benefits to immunizing at birth, and this is the mechanisms for this remain yet to be determined. So no clear downside to immunizing with BCG at birth for TB protection, and potentially even advantages. But the main point of our talk today wasn't to think of BCG as a vaccine that targets tuberculosis, but targets many other pathogens, so a pathogen agnostic or host-centered approach. And here it is absolutely crucial to understand that if you don't use BCG at birth, you miss the big window of vulnerability, because newborns die in the first few days after birth. And that really is the main impetus that drives immunizing newborns, or immunizing with BCG at birth.
And one last question is, Nelly commented on how metabolism plays a very significant role in outcomes. So what might be considerations for ramping up or making available energy sources for newborns who perhaps are going in the wrong direction in terms of their ability to mount an immune response?
Yeah, well, this is exactly, pretty much exactly where the next wave of our research is going to be focused, because there's this beautiful intersection around the non-specific effects of BCG, but what has been equally as ignored in newborn immunology, and especially in our mouse modeling, is nutrition, which for newborns is their feed, but it's the timing and the type. Of course, that differs by gestational age as well. But we know also from epidemiological studies, and this data comes out of Ghana, that if you delay the initiation of breastfeeding by a few hours, you can double the risk for death by infection in those kids. But of course, that's an epidemiological association. We know that there are a lot of benefits that colostrum offers. And one of our collaborators here, Valerie Verhasselt, has been researching breast milk for over a decade, and is now finding how colostrum is really irreplaceable, even if you have donor human milk. So donor human milk in the clinic is better than formula, but it actually doesn't seem to provide the same protection from infectious diseases. So what we actually have no clue, and I think there was a larger picture in the a larger post in the chat around there are other challenges in the field around why newborns get sick and why they die, in terms of what kind of access do they have to healthcare, whether they're born in remote settings, breastfeeding practices. We don't know how breastfeeding practices modify the effect of the BCG vaccine or any vaccine in the first few days of life. No one's actually tested that. And one of the first things that we thought of is exactly that. Immunity is expensive. Now, we know that from adult literature, and a great review came out last week, it was by Yasmin Belkaid's team, it was about nutrition and immunity. And in the fasted state, you can suppress inflammation, but without inhibiting the ability of neutrophils to clear bacteria and stuff like that. So it is possible to have a pool of neutrophils in a newborn that isn't going to cause harm, but there still has to be adequate fuel, and in order for the expansion to happen, and in order for tissue damage to also not occur. So we don't know. The short answer is no one has actually tested it, which is why we would love to get on this immediately and actually put this to the test. There are ways to get at this in a laboratory setting, and of course it's really hard because there are a lot of things that we will just always fail to recapitulate in an animal model. The beautiful thing about this research so far is the low birth weight trial that first showed a benefit of BCG was based on observational research, that was really taken from a demographic surveillance system in Guinea-Bissau. So this is really out in the field observations. These weren't kids that were closely monitored in a hospital. This was data that was recorded on small sheets of paper and in follow-up visits. And this is where the effective BCG was first found. And when it was tested in a randomized trial, it was actually amongst healthy babies that were then sent home from the hospital and at home with their mothers. And then some of them would get sick in the next few days and die, and others wouldn't. So again, this effect was found in the community, and this is how we found out about it. And then we went into our mouse models to figure it out. So now we're doing the same thing with feeding. We're going into the literature, seeing what have we found in epidemiological studies around newborn nutrition that seems to matter, and how could we model this in a way that at least kind of makes sense. But it's always on us to then go back into human systems and see if what we find actually matters. But yeah, that's exactly it.
Thank you. And with that, I'm afraid I'm going to have to cut you off. But we really appreciate obviously the passion that you both have, that you bring to this, and thank you for sharing with us. I'd ask if you could go into both the chat and the Q&A and answer some questions. Noted that you're looking for students and postdocs. So again, we invite people to go to your emails as you posted. And thank you very much for that very informative and stimulating discussion point about newborns and not the pathogen, but the host. And with that, I'm going to turn over to Professor Nadia Hamdi, who's now going to introduce our next speaker. Thank you both very much.
Thank you.